of the strands of DNA look like. This is like a close-up. It's made of a phosphate group, a sugar, so ribose for RNA and deoxyribose for DNA, and a nitrogenous base, and in this example it's thymine. And so the yellow and the blue make up the sugar phosphate backbone, and then the nitrogenous base changes with each base pair, and that sticks out and bonds to other nitrogenous bases. So that segues into the difference in the sugars between DNA and RNA. So for DNA, it's deoxyribose, and that's a diagram right here. And then for RNA, it's ribose, so that means there's an added OH group in blue. And then they're both covalent bonding, and DNA is found in the nucleus, while RNA is found in the nucleus and the cytoplasm. In this slide, we're going to talk about how a specific base pairing of a DNA's nucleotides dictates DNA replication. So in over here in the first diagram, we just see that we have the parental DNA. So the parental DNA strand is going to split, and each half is going to become a template for duplication. And one of the challenges in DNA splitting is that the double helical shape really adds complexity. So because it's helical, it's hard to split because it has to unwind and it has to copy simultaneously. So it's pretty difficult. So then after it's split, we see that a free supply of nucleotides come in and base and pair up with the corresponding base, with, you know how A and T come together and C and G come together, and certain enzymes link the free nucleotides to the template strands, and once they're linked, we have two new completed models, which are called daughter molecules. So DNA replication begins at places called origins of replication, which is this diagram here. So origins of replication are certain places or certain sections of DNA that have a specific sequence of nucleotides that determine the function and where proteins attach and how the strands separate. And many sites allow for a more efficient replication process. So basically, the parent DNA strands, which are the pink ones, open as the daughter strands elongate on both sides of the bubble. So the process occurs in both directions, and at the very, very end, all of the bubbles merge, yielding two completed daughter DNA molecules, which are supposed to be each of these. So there are actually more than a dozen enzymes involved with DNA replication, but I'm just going to talk about two of them. So the first one, DNA polymerase, or I'm not really sure how to say it, is an enzyme that links DNA nucleotides to a growing daughter strand, and it adds nucleotides to only the 3 end and allows growth in the 5 to 3 direction. So the arrow down at the bottom right here is just showing that it's moving in the opposite direction, and the enzyme is the blue, which is you can see is attaching. And then the DNA ligase, which is this bottom enzyme, which is this green one, links pieces to form a single DNA strand. And there's also a special function of enzymes called, well, they basically proofread. So they remove incorrect base pairing of nucleotides, and they repair damaged DNA from harmful radiation and toxic chemicals. So in this slide, we're going to talk about the differences between transcription, which is blue, and translation, which is pink, in terms of location of each of these stages, reactants involved, and the products produced. So transcription is the changing of DNA to an RNA molecule in terms of genetic information, and then in terms of genetic information, translation is the transfer of RNA into a protein, and the location of transcription is the nucleus, because that's in fact where the DNA is located, and translation occurs outside of the nucleus in the cytoplasm. And the reactants of transcription are free nucleotides that attach to form an mRNA strand, from the DNA template, and then translation, the reactants are the RNA produced from transcription, which would be further changed into the protein, and then the products of transcription are mRNA, tRNA, and rRNA, while the products of translation are a protein that was encoded for. And over here we have a diagram just of the basic sequence. So we have DNA that's changed into RNA through transcription, and then that occurs in the nucleus, and then translation occurs outside of the nucleus in the cytoplasm, where we get the protein as a product. So in this slide we're going to talk about how the DNA and RNA languages are used for polypeptide production. So right here, this is just a general overview of the movement between DNA to RNA through transcription, and then from RNA to a polypeptide chain through translation. So basically, we are right here, the nucleic acid language of DNA has been rewritten as a sequence of bases on RNA, and the language is that of nucleic acid. And just to really quickly talk about what languages actually mean, it's basically just a form of communication between the parts of DNA and RNA that change DNA to RNA to protein. And so then right here is the most important step, which is the results of translation, which changes the nucleic acids into the polypeptide chain language. And polypeptides are polymers, but the monomers are just the 20 amino acids. And the language is written in a linear sequence, which is really important. And the sequence of the nucleotides, it's what directly influences the sequence of amino acids. So, the flow of information from gene to protein is the triplet code, which is the instructions for the amino acid sequence of a polypeptide chain are written in the 3 base words called codons. So that's right here. So these 3 base nucleotides are directly influencing which shape is created. So that's just showing how this specific arrangement of the nucleotides directly contributes to the polypeptide production. So in this slide we're going to talk about the production of RNA, which is called transcription, and it occurs in the nucleus because RNA is transcribed from DNA, which is, as we know, held in the nucleus. So the process begins in initiation. So basically the 2 DNA strands separate and one is used as a template, and then an enzyme, RNA polymerase, attaches onto a specific sequence called the promoter, and when it attaches, basically a signal is sent out which says start transcribing and start creating this RNA chain kind of, and it also helps to decide which of the 2 strands of DNA will be used. So that occurs at this green area called the promoter DNA. So in the second phase, which is called elongation, the RNA synthesis continues and the RNA strand peels back from the template, so the 2 other strands of DNA, or the 1 other strand, comes together with the first. And then in the third phase, called termination, the RNA polymerase, so the enzyme, reaches a sequence of bases called the terminator, which is shown in the red, and that basically says stop because you're at the end of the gene, and the enzyme detaches from the RNA molecule and the gene. And right here is just a close-up of the transcription. So as you see in the 2 blue, these are the 2 strands of DNA, and the pink are the RNA nucleotides that are attaching to form this new strand of RNA. And the transcription is moving in this direction, and as you can see, the 2 DNA strands come back together in the end. So in this slide we're going to talk about how RNA is processed before leaving the nucleus. So basically, the type of RNA that encodes amino acid sequences is mRNA, which stands for messenger RNA. And it is transcribed from DNA, and then the message which is encoded in that mRNA is used to create polypeptides in the cytoplasm. So we know that before the mRNA leaves the nucleus, it actually is transformed in a couple of ways, and in this picture we see one of the ways. So in this picture we are adding extra nucleotides to either end, so that is the cap and the tail. And the cap is just a single G nucleotide, while the tail is 50 to 250 adenine nucleotides. And this addition helps to facilitate the export of mRNA from the nucleus, and it helps with ribosome bonding, and it helps protect the mRNA from attack, and it's not actually translated to protein, so it's just there. So in addition, eukaryotic cells have to have another type of RNA processing because the sequence that codes for the production of polypeptides isn't actually continuous. So we have these two regions, these two internal regions called exons and introns, and exons are the coding region and they're expressed as an amino acid, while the introns are the internal non-coding region. So as we see during splicing, RNA splicing, the introns are removed because they don't serve an actual coding purpose, and the exons are spliced together to form one continuous coding sequence. And to actually get the splicing to occur, it's catalyzed by a lot of complex proteins and small RNA molecules, but sometimes it's actually catalyzed by the RNA transcript itself. So in this section we're going to talk about how the structure of tRNA relates to its functions in the process of translation. So the structure of tRNA, which stands for translation, is just a single strand, and it's made of approximately 80 nucleotides which coil and form double-stranded regions where the stretches of nucleotides pair to each other by hydrogen bonds, which we can see here. So there's this one strand of polynucleotide chain, and it's folded, and each of the bases pair to each other by base-specific rules. And actually there's this special thing that's formed, which is a triplet of bases called the anticodon, which has the unique feature of being complementary to the codon triplet. So it recognizes the codon on mRNA with base pairing rules, and then on the other end is a site where amino acids can attach with specific enzymes. So basically, a cell has a cytoplasm that's full of amino acids from foods or chemicals, and the amino acids can't recognize codons in the mRNA first, so they have to pick up the appropriate amino acids and recognize the appropriate codons in mRNA, which is allowed by this special structure formed called the anticodon.